To begin with, in session 4, we are delighted and very honored to have Dr. Lucas Amenga-Etego from University of Ghana joining us to talk about genetic surveillance of malaria in Africa. Now, just as a way of introduction, Lucas has been a long time friend of Malaria Gen, and he is conducting a project in Ghana, which is very similar, both in objectives and in technologies, to what we're doing in Southeast Asia with GenRe-Mekong. So what I hope he'll be able to give us a view on how that is proceeding, and perhaps how in the future our projects can work together. Lucas, are you there?
Yes, Oliver, I'm here.
Good, welcome. Take over, please.
Can you guys see my slides?
Yes, thank you.
Thank you, Dr. Oliver and all the organizers. Thank you for the opportunity to share from my perspective how genetic surveillance of malaria is doing in Africa. I know there are several people who could have given this talk probably much better, but I thank you for settling on me to contribute to your meeting. I'll start by acknowledging our local team and partners who have been supporting my work in GSM, in Ghana here, and across our other partners in Africa. Particularly, I want to mention Dr. Kezia, who is our National Malaria Control Program Manager. She's been very supportive of our work. And of course, the workhorse in our labs is Joyce. And we're very grateful to the Sangha team for supporting us through all our challenges. Particularly want to thank Savanti, Vicky, Dominic, everybody in the team, informatics people. We are grateful for your support. My talk, I've structured my talk in a way that I would want to start by giving you an idea or a sense of how malaria and the landscape is across Africa, the kinds of things that we think we can do with GSM, the genetic threats that we can begin to throw and shed light on using GSM technology, and the current situation as far as anti-malaria drug resistance is, and a little bit on insecticide resistance, and some of the mixed bag of use cases that I think, from my perspective, I think various countries would want to adapt and increase or expand as GSM takes root on the continent. Also, GSM capacity and the current key players that I am aware of, I would highlight some of those focal studies going on, and talk a little bit about stakeholder engagements and the challenges for sustainable GSM in Africa. And then I would conclude and give you some future perspectives. So malaria transmission, vector landscape, and the parasites quite heterogeneous across our continent. We do have perennial transmission and across sub-Saharan Africa, but there's also marked seasonality as you go from west to east, and that is important in the way gene flow is assessed. And we do have quite a good number of vectors that are very competent. Anopheles Kuluzy, Fenestus, these are quite popular in Africa, and Arabiensis. There's a mixed bag of others that, depending on where you are on the continent, you'll have to contend with probably 3 or 4 different very competent, equally competent vectors And so that affects the way transmission and prevalence of parasites vary across our continent. So the question I ask is, how could GSM, or genetic surveillance of malaria, help us to better present these maps in a way that we can highlight the risk that different products would help us to understand going forward when we are able to expand GSM in our continent. So within our continent, we do have, just like in many other places, different drugs being used, but there's a lot of commonality. As you would see, our frontline drugs in these example countries, you can see AL, tetramethylamine, DHA, piperaquine, all these are quite popular in our continent, and we expect that we can share data that we generate regarding some of the genetic threats that we would probably see that are arising against these frontline medicines. There are also a lot of interventions. Some are targeted, like the seasonal malaria prevention, that is targeted in areas of high transmission that have seasonality, like it happens in West Africa, where children are given preventive treatment entering the high transmission season. And these need to be monitored to see if these drugs are imposing a little more selective pressure on the parasite populations. There's long use of insecticide-treated nets, and IRS is going on. National malaria control programs are trying to drive down malaria, indoor residual spraying. There's also malaria prevention in pregnancy, that's using SP. These are all interventions that across Africa are happening as we speak. And SMC, which is not happening in all places, but is key because it is targeting children under 5, and of course imposing extra pressure. We would want to see what would happen with the parasite populations going forward. So what would GSM help us? How is it going to help us to properly gauge the effect of these interventions, and also measure their impact in a way, sort of trying to, monitor and see what would happen post SMC, and also what the implications are for malaria control. There are myriad of vectors, as I said, that are across our continent, but a lot of them are common. Anopheles gambiae is a competent vector across our countries. There's Anopheles fenestus that's occurring many places, but there are some unique countries that have unique vectors, like we have in East Africa, Tanzania, Ethiopia. And so it will be interesting if GSM takes roots in Africa, and we can compare our data to see what we get out of the African landscape. So the potential use cases, we will want to fight genetic threats. We want to look at the risk for antimalarial drug resistance, diagnostic failure due to HRP2 gene deletions are also important in our toolkit as Africans, because as I said, transmission is perennial, and so it's important that we are using our frontline drugs effectively to treat people who are actually having malaria, and parasites can be detected accurately. And as you may be aware, the WHO threshold is a prevalence of 5% for you to change your RDT. And so given the fact that there are not really available alternatives, it's important that we keep monitoring and ensuring that we're keeping an eye on that prevalence. We have been using insecticides to treat our bed nets for a very long time. That intervention has several decades, and there is indoor residual spraying that's using various classes of insecticides, and farmers are using insecticides as well. So it is no surprise that insecticide resistance in our vector mosquitoes would arise. And so it's important that we generate data that can be used to monitor mosquito populations and see what kind of resistance that we can assay. Also, in general, parasite population genetics, looking at the population diversity of parasites within borders, like we do in Ghana here, trying to use GSM to see what is the landscape of resistant mosquitoes or parasites, Plasmodium parasites in our borders. And we can integrate our data to look beyond our borders into the regional and possibly in the future extend to other parts of the continent. And for areas in East Africa where there is low transmission, I reckon they can begin to look at importation of specific parasite genotypes between defined areas, and also generally see how interventions are disrupting the parasite population structure. So what is the current situation in Africa regarding anti-malaria drug resistance? CALT stage 13, since it arose in Southeast Asia, in your region, and we're all aware of the historical spread. So the danger is Africa needs to prime itself to see what happens going forward. And in 2018 in Rwanda, CALT 561 was identified. It's been gradually increasing the frequency in Rwanda, but you look at the clinical cases and treatment outcomes, and the fact is we still haven't seen marked ACT resistance. They remain effective in Rwanda and also other parts of the continent. Several independent detections of specific CALT13 mutations like the R622I mutation in Africa, the Horn of Africa, Sudan, Somalia. These have been detected and they need to be monitored, but as of now, we don't see them altering treatment outcomes to our frontline drugs that I mentioned earlier on. But the absence of resistance simply tells us that we need to keep an eye on our frontline drugs. We need to continue to monitor, and GSM comes in handy for us to be able to do this with greater depth and the opportunity it gives us to be more accurate in determining these resistance alleles going forward. The partner drugs are important, not just artemisinin, and you saw from our previous slides that there are quite a number of common partner drugs that are being used. Chloroquine is not completely gone in Ethiopia. They will use it to treat vivax. In Madagascar, they use it to treat vivax as well. So it's not, we are not getting a complete expansion of chloroquine sensitive parasites in Africa. And there remains some focal areas where chloroquine resistance is important to monitor. Also, in terms of vectors, our use of pyrethroids for many years has led to significant resistance against pyrethroid insecticides that we use to control mosquito vectors, particularly the KDR L995F allele, which is very dominant in West Africa, spreading towards central and of course central southeast direction. This is an important mutation that is quite prevalent in West Africa and sort of undermining pyrethroid, that class of insecticides that we use as aerosols at home or treatment in our bed nets to prevent mosquitoes from getting access to us as we sleep. And there are other SNPs in that region of the VGSC gene, sodium voltage channel, beta channel gene that is a target gene for pyrethroids. And these need to be monitored because there's evidence from Clarkson and his colleagues that when certain haplotypes when they occur with the 995F, they do increase resistance significantly. And as we're moving towards adding PBOs to our insecticide-treated nets together with pyrethroids in order to try to improve their efficacy, no one is sure what the future holds for PBO-treated nets. So we need to keep an eagle eye on the process and continue to monitor those nets to see what benefits PBOs can impose on long-term treated nets, and if the benefit that we hope to achieve by combining these insecticides will stand the test of time. So I mentioned the VGSC gene. This is a voltage gated channel involved in the proper function of the mosquito nervous system. And as you can see, there are 4 domains, and these domains are critical for monitoring in the resistance, because as the L995 locus here, as you can see, is key in resistance, and there are myriad of them, haplotypes formed over there and there. And so it's important for us to, not lose sense of the fact that the additional PBOs may distort the current patterns that we're seeing in terms of the SNPs and the mutations that are occurring in this gene. And so I'll just show you a little bit of what is happening in Ghana. Specifically, you can see the L995F that I was talking about. It's 100%. It's fixed. The mutant allele F is fixed across my country, but from the south, the north, and the middle belt of our country is totally fixed. And you can see a whole lot of mutations that may potentially form haplotypes with this mutation and cause increased resistance to GAMBI. But when you come to Anopheles Kuluzy, you can see that the resistance to Kuluzy for the same locus is not fixed yet, but it's high, and it's highly variable across different parts of our country. This kind of monitoring is critical for our NMCP, because then it allows them to think about how to shuffle their insecticides and also plan going forward, making decisions as to which parts of the country they should target with what insecticides. So the key players, as I said, to my knowledge, in genetic surveillance of malaria in Africa are members of the PDNA, who is a group of researchers that have been working together for many years, led by Professor Jimdi. And in Ghana, we do have a GSM project, like Oliver mentioned. And we're doing this in collaboration with Alfred Amagwa in the Gambia. And of course, Anita is in the Bush Memorial Institute of Medical Research in the University of Ghana. She also looking at GSM, and we are complementing each other, working with our NMCP to see how we can drive GSM in our country and use it to help the NMCP to make informed decisions about insecticides and drugs. Also, in East Africa, I know Deos and his group, working with his partners in the US and in Sanga, are looking at employing GSM to monitor their parasite populations. The Tanzania has a wide heterogeneity in transmission, and so they have the opportunity to do a lot of things, expand their use cases to be able to understand various aspects of their continent. I know there are several others that could be doing things in the continent, but I don't kind of say for sure. And so these are the key players in our continent at the moment. And what are the tools that are gaining traction? We are using the amplicon sequencing approach, and I know the Gambia, Mali are considering the same approaches, but in Tanzania, molecular inversion probes are being tested, to look for variation of Plasmodium falciparum. And so there's the opportunity in our continent for people to either use both approaches or one or the other. And the opportunity still exists for us to do whole genome sequencing approaches. As you may be aware, the tools are at different stages of development. We do have better understanding, more working tools of the amplicon sequencing toolkit than we will have for assays that we want to do on the insecticide or mosquito side of things. And so with our mosquitoes, what we're doing is trying to do whole genome sequencing for mosquitoes and then do amplicon sequencing for Plasmodium falciparum, and try to look at population studies, look at drug resistance, insecticide resistance with whole genome sequencing, HRP2 deletions. They are very critical for our NMCP, as the WHO is pushing for African malaria control programs to try to understand the prevalence of these deletions in their areas of jurisdiction. So stakeholder engagement is critical for the success of GSM in Africa. We do have relationships between our NMCPs, but these remain quite fragile. The NMCPs fundamentally do not usually, have technical understanding of the data. The data is quite challenging both for researchers and NMCPs, and so we, those who are in the GSM space in Africa, need to find creative ways to build bridges with the NMCPs, try to sustain their interest, and try to see how we can support the NMCPs to appreciate the products that we would be turning out, and help them to see how they can integrate our work into their daily work and influence decision making. In our case, my observation is where the NMCP managers have a scientific background, this relationship becomes a little bit easier. There is better understanding, and then there is room for discussion, and we can work together to formulate use cases that are most beneficial to them. So there's a need to continue to engage NMCPs, and across Africa, people are using various approaches that are custom to their environment to try to test out how best to sustain the interests of the NMCPs. The point is, the NMCPs in most countries have selected health facilities that they call their sentinel sites, that they use to sample and probe the effectiveness of frontline compounds. And so it's in our interest to ensure that our sampling that we do for genetic surveillance does coincide with some of these facilities that they are covering in their daily work, so that data can be easily translatable to their decision making. And the only way to achieve that is when we have a close relationship with our NMCPs. So we're trying to work on that fragile relationship and strengthen it going forward, and hoping that we can achieve a sustainable GSM. In the light of this, our NMCPs in Africa need reorientation and training. They do have technical people working with them, and as I said, the complexity of the data we hope to present to them requires that they build some technical understanding of the basics of GSM and the products that we would be sharing with the NMCP. So basic skills and a changing mindset. The NMCPs look at researchers as people driving this initiative, but I think going forward, it's about time for the NMCP to take center stage in trying to influence what we do, how we define our use cases, what we need to do that benefits them the most. And so it's delightful to have Professor Jim and Paulina getting a grant from the Gates Foundation to engage the NMCP across Africa to begin to build their capacity to sort of tailor their needs for GSM, and see what kind of interventions they can put in place that will help them to appreciate the data and also strengthen that relationship that our NMCPs have with scientists that are working in the space of genetic surveillance of malaria. So it's a welcome grant that we hope would impact the NMCPs across our continent, and then increase our engagement and lead to positive outcomes going forward. The NMCPs, in my view, currently look at GSM as something that researchers are pushing them to adapt, and I think that perspective is not good for sustainability. They need to understand that it's a shared responsibility, and I'm hoping that the grants that Professor Jim Lee is holding would help us to push this agenda and cause a change in mindset in our NMCPs that will allow them to begin to appreciate and take interest in technical skills that are relevant for their understanding of the data. So in my view, in Africa, what are the challenges? Several platforms are now being introduced in Africa. Key among these is the MySeq that we are using to operationalize the amplicon sequencing toolkit in Ghana and Mali and other places. It's quite a daunting task to have maintenance contracts for these machines. Region procurement, to say the least, is completely a ripoff for us as scientists, because we pay an order of magnitude much higher for our regions, and we have to wait for a very long time to receive them. It's a bottleneck for GSM in Africa that needs to be addressed, and the capacity to understand the data is something that is building up slowly across our continent. Surely, I believe going forward, GSM activities would lead to an increased critical mass of people across Africa who have a knowledge of the data and can translate this to products that the NMCPs would appreciate and find useful for their day-to-day decisions. Unfortunately, we have poor infrastructure in terms of computing for data storage and processing. There are not many places where there are servers that we can use to run this data, and internet bandwidth is a problem. There's power instability. I mean, there are quite a number of things that are challenging for our environment, but they are not in any way unsurmountable. Over the last 3 years, we have improved our infrastructure. We have particularly, taken note of these and work towards, creating the right environment to allow GSM via the amplicon 2K to work. And I am pretty confident that we will begin to generate data very soon, and that would be exemplary for other African countries to follow. The Gambia is doing the same thing. They are in a much better position than we are to deal with these challenges, and I'm sure we would have knowledge generated from there that we can share across our GSM partners and understand how to handle these challenges going forward. And so, to conclude, there is an opportunity for the NMCP to grab. GSM is come to stay, and there are clear benefits for doing GSM in Africa. If there's nothing at all that we can learn from the COVID-19 pandemic, has given GSM a lot of momentum. Molecular surveillance is now a key word even among politicians, and I think the NMCP need to seize the opportunity to work with scientists and make GSM a reality and a practicality on the ground. Capacity exists for this to be done, and there's a need to improve it across sub-Saharan Africa. We cannot develop processes and means to do pandemic surveillance when there's a need, and then we ignore endemic surveillance. The problem is we need a complementary approach to ensuring that these are given attention where there is a need. In a country in Africa here, we would need to have the same attention that was given to COVID to be applied to malaria and other endemic diseases, and we can have a surveillance system that is sustained to handle all scenarios of pathogen behavior in our continent. African NMCPs need support. They need real support from donors to be able to utilize the data. They need to orient themselves. They need to gain technical understanding of the data, and so that cannot come without cost. It has cost implications, and I'm glad that some funders are beginning to address this issue. Equitable access and to equipment and reagents, as I mentioned, is important. I don't know how this is going to happen, but we need to take steps to ensure that there's equitable access to reagents in particular to allow GSM to be sustainable in our continent. Sorry, repeat of that. So I'd like to end here and highlight some of our funders, particularly the National Institute of Health UK, who are sponsoring us. They are giving us money to build infrastructure and allow GSM to work in Ghana and the Gambia, and hopefully our story would be something that can be expanded to include other African countries. And of course, Mali is poised to take on this task, and of course other countries in Central Africa that are key to adopting this technology, and we can have a drive from West, Central to East to ensure that this works. We thank our Malaria partners. We thank the Wellcome Trust and the UK Aid for their being supporting our lab. Thank you all for listening. I'll drop it here. Thank you.
Thank you, Lucas, for this great and very comprehensive talk about surveillance in Africa. I should just add a shout here for another partner in Africa. There's in the Congo, we have the Kimoru site, who are and Dr. Caterina Fanello is in this call, and they are they're currently running a study to use on whether a cohort of pregnant women presenting at maternity clinics can be used as a surrogate for doing surveillance instead of using small children, which of course are a more problematic population. So, and what we're hoping is that we'll be able to move forward with implementing in that country also. And what I found was encouraging is to see that many of the issues that you discuss there, such as capacity building, training, empowering the national malaria control programs to better receive the data and better interpret it, are common themes. And I'm really hoping that we'll be able to solve these things together, maybe by some joint training and so on. So I'm sort of putting that out there for discussion later. Lucas, you have a couple of questions or comments from Sanger Institute here, from Roberto. He says, thank you for this great eye-opening talk, Lucas. Whole genome and amplicon are clearly complementary technologies, but your point about the complexity is really, really important. How do you think the trade-off between these two approaches should work? Would you like to answer that, Lucas?
Yes, please. I thank you, Roberto, for that question. Well, I think for a much wider implementation of GSM in Africa, I would advocate that we go through a more targeted approach, like the amplicon sequencing toolkit or the MIPS that other people are doing. But as you said, it's a complementary approach. And so we will still need to build upon the targeted sequencing with whole genome sequencing, where we take advantage of knowing, increasing the breadth of the data to be able to answer some of the population level questions that will become increasingly important as we drive down malaria transmission across the continent. Thank you.
If I could just add one comment to that, our team, when investigating the outbreak in Laos, found it very useful to be able to do initial analysis using amplicon sequencing, using genetic barcodes, in order to sort of understand and characterize the problem. And then we were able to dig in a lot deeper with whole genome sequencing, for instance, looking at the ancestry of these R539T mutants that have caused the outbreak. So I think I see a really great motivation for actually implementing different technologies. One doesn't replace the other. But of course, it is a big problem. How do we make it accessible to everybody? And right now, I think we have to go for a while in relatively small steps, because the first task is really to make even the relatively simpler methods more accessible to the countries, because right now it is still quite difficult to use the data, even the genetic report cards, even the sort of more simplified data that we can get from amplicon sequencing. We really have to work on training and focus on how to empower the control programs and the scientific partners to make full use of these. And then I think we'll be able to approach the whole genome sequencing. Richard Pearson is asking another question to Lucas, which I suspect is somewhat related here. Says, what do you think is the key to increasing the data analysis capacity in Africa?
Well, thank you, Richard. I think the key, in my view, is hands-on training. It's more targeted training that people who are generating data and can train and engage their data would be, in my view, in a better position to understand and use the skills. Over the years, I've learned that people do training and go back and they lose their skills because they don't have data to engage. But when there's data being produced and there's capacity building, data analysis skills are being transferred, then I can foresee a situation where the skills are being built and honed in a way that there's immediate value for the skills, and they can be a trainer of trainers. And so you get a lot more people benefiting from one person gaining key knowledge and using the opportunity that exists within his space and with the presence of data to be able to train other people. So my take is that these cannot be done on a theoretical level. This cannot be done in a situation where there's a one-off training and people are asked to get up and hone those skills. No, we have to do this in the context of data availability, a context of progressive and continuous build-up in people's capacity. Thank you.
Lucas, I think that's incredibly valuable insight. Thank you very much for that. I think, I think you've crystallized some of the things that we have been really thinking about doing some training. That it has to be accompanied by data, it has to be accompanied by real problems that can be practiced upon. There's a question here from Sasha, is on something different. She's asking whether the whole genome sequence data for the vector insecticide resistance surveillance, whether it's enough to begin implementing targeted approaches, or are our known markers for insecticide resistance not mature enough yet? What's your thinking?
Well, my thinking is, as we've said previously for Plasmodium, it's the same approach for the mosquitoes. The whole genome sequencing and the target approaches are complementary, but the toolkit, getting the markers that target genes in the mosquitoes that have been sort of phenotyped for you to know for sure that they correlate with a specific resistance to a specific class of compounds, still is a bit of a gray area. We need to first of all generate data that validates all the SNPs that we see, say in the VGSC gene, and gather the kind of phenotypic understanding of how these mutations translate to resistance, and then build targeted panels that can capture these mutations and generate data that we can use to monitor, while still doing whole genome sequencing for mosquitoes, vector mosquitoes, to understand in greater detail the population level statistics, and also just general diversity and selection as it happens in other genomes. Thank you.
Thank you very much.